Your body is this incredible universe made up of trillions of tiny cells. And the process that lets you grow or heal a cut on your finger, it's all down to this beautiful, microscopic dance called cell division. It's really the engine of life. But what happens when that engine just refuses to shut off? Let's get into it.
Okay, so normally, cells have a built-in expiration date. They divide a certain number of times, and then that's it. They stop. It's one of the fundamental rules of biology. But in science, it's the exceptions to the rule that often lead to the biggest breakthroughs. And back in the 1950s, scientists stumbled upon one whopper of an exception.
I want you to meet Henrietta Lacks. Back in 1951, cells were taken from her cancerous tumor, by the way, without her knowledge. Now, unlike any human cells ever seen before, hers just didn't die in the lab. They kept dividing, and they're still dividing today. These HeLa cells, as they're called, became one of the most important tools in modern medicine, helping with everything from vaccines to cancer research. But they also left us with this huge mystery.
To get to the bottom of this, we can't just stare at this one immortal cell. No, we've got to understand the normal, super-regulated process that governs the life of every other cell in your body. We need to get a feel for the fundamental rhythm of life.
And that rhythm, it's called the cell cycle. And it's not just a cell splitting in two, not at all. It's this carefully choreographed series of events that makes sure that when one cell becomes two, each of those new daughter cells has exactly what it needs to survive and do its job perfectly.
The main thing to get here is that the cell cycle is anything but random. It's a tightly controlled process. Think of it like a cellular assembly line, with really strict quality control managers at every step. To figure out how HeLa cells break all the rules, we first have to understand what those rules are.
So if you look at the whole cycle, it really breaks down into two major stages. You've got the M phase, or the mitotic phase. That's the actual, division part. But look at that huge chunk of the pie chart. That's interphase. And that's where all the real prep work happens.
And I'm not exaggerating. A typical human cell spends about 95% of its entire life in interphase. That dramatic split that we always see in textbooks, that's just the grand finale. The vast majority of a cell's existence is just getting ready for that one big moment.
So let's zoom in on interphase. Now, people used to call this the resting phase, but that could not be more wrong. This is an incredibly active time of growth and, most critically, copying the cell's most precious possession, its DNA.
Interphase itself is broken down into three smaller steps. First up is G1 or gap 1. This is where the cell bulks up and just does its normal day-to-day job. Then comes the really critical part, the S phase, which stands for synthesis. And finally, there's G2 or gap 2, which is like the final systems check before launching the division process.
The S phase is really the point of no return. This is where the cell painstakingly duplicates its entire genetic code. Every single one of the 3 billion letters of its DNA is copied to make a perfect, identical second set. It has to be perfect, so each new cell gets a complete instruction manual.
Now here's a detail that trips a lot of people up, but it's super important. After the S phase, the amount of DNA in the cell has literally doubled. It goes from 2C to 4C. But the number of chromosomes, 2N, technically stays the same. How? Well, it's because those copied DNA strands, called sister chromatids, are still stuck together, forming one of those classic X-shaped chromosomes, until the very moment they get pulled apart.
It's also worth pointing out that not every cell in your body is on this constant track to division. Some, like our mature brain cells, actually leave the cycle entirely and just hang out in this quiet stage called G0. They're still alive and working, but they've pretty much retired from the division game. It's a big reason why injuries to the brain and spinal cord are so hard to fix.
Okay, the cell has grown, it's copied all of its DNA, and it's passed all the final checks in G2. So now it's time for the main event, the M phase. This is where one cell physically splits into two in that amazing process we call mitosis.
First up is prophase. All that long, spaghetti-like DNA that was copied earlier now coils up on itself like crazy, condensing into those thick, visible chromosomes you recognize from biology class. At the same time, this structure called the mitotic spindle starts to form. Think of it like a set of microscopic cranes ready to move the heavy cargo. Next, in metaphase, those condensed chromosomes get grabbed by the spindle fibers and dragged so they line up perfectly, single file, right down the middle of the cell. This lineup on what's called the metaphase plate is absolutely crucial.
It's the cell's way of making sure that when the split happens, each new cell gets exactly one copy of every single chromosome. No more, no less.
And then comes the most dramatic moment of the whole show, anaphase. In an instant, the connections holding those sister chromatids together dissolve. The spindle fibers yank them apart, pulling the identical copies, which are now considered their own individual chromosomes, to opposite ends of the cell. It's this beautiful, coordinated tug-of-war.
In the final stage of mitosis, telophase, the chromosomes finally arrive at their destinations at opposite poles. They start to unwind and decondense, and a brand new nuclear envelope forms around each of the two complete sets of DNA. The nucleus has now officially divided in two.
But wait, we're not quite done. The last step of the whole process is cytokinesis. This is the division of the rest of the cell, the cytoplasm. And this is where we see a really cool difference in biology. Animal cells, because they're soft and squishy, just pinch in the middle until they pop into two. But plant cells with their rigid cell walls can't do that. Instead, they have to build a brand new wall called a cell plate right down the center. Two totally different strategies to get to the same end goal. Two brand new independent cells.
So that's the normal, neat, and tidy process. But because it's so incredibly complex, there's always a chance things can go wrong. And to prevent that, the cell has this amazing internal control system. And understanding that system is what's going to bring us right back to our original mystery.
All throughout the cycle, there are these critical checkpoints. Seriously, think of them like bouncers at a club. At the G1 checkpoint, the cell asks, am I big enough? Is the environment right for this? At the S checkpoint, it's got to confirm, did we copy the DNA correctly with no mistakes? And then, during mitosis, it has to check, are all the chromosomes lined up and ready to go? If the answer to any of those questions is no, the whole process slams to a halt.
So what happens when that control system breaks? What if a cell's DNA gets damaged in just the wrong way, affecting the very genes that run these checkpoints? Well, the cell basically loses its brakes. It starts dividing without control, ignoring all the stop signs. And that, right there, is the fundamental definition of cancer.
And that brings us full circle back to Henrietta Lacks. The secret to her cell's so-called immortality wasn't some kind of magic. It was a tragedy. Her cells were aggressive cervical cancer cells. They had suffered so much genetic damage that their cell cycle checkpoints were completely and utterly broken. They were biologically incapable of stopping. A trait that, while devastating for Henrietta, made them priceless for science.
So when you think about it, the very same system that allows a baby to grow, a wound to heal, that same exact system is just a few broken genes away from the total chaos of cancer. The story of the cell cycle isn't just about how cells divide. It's about the incredibly delicate, fragile system of control that makes complex life possible in the first place. Kind of makes you wonder about that fine line between order and chaos in all of biology, doesn't it? Something to chew on.